So welcome back. I'm sorry for the network glitch. We are talking about the absorption, and we did mention that absorption of these xenobiotics depend on the route of exposure. And we basically mentioned some route of exposure, most especially in the case of drugs. We can have drugs that are meant to be taken orally. You have drugs that are meant to be uh, taken maybe through inhalation, like the asthma drugs and some others, which basically use an inhaler. You have some drops that are meant to be applied on the skin, more or less of topical application. And you have those that are injected either intravenous, intramuscular, and so many others. So when we basically look at the most common one with respect to drugs, but because if we are talking about the pollutants or food additives, for example, you food additives, in most cases we talked about oral ingestion, because you eat food or you drink water, which will have to pass through the GIT, and then the absorption takes place around the intestine. So while in the case of pollutant, you can have some of this pollutant being part of the food where you ingest, or you have this pollutant being part of the maybe in gaseous form where you inhale them, or you have them as a dust that can attach to your skin and then get into the body. So let's look at, we are talking about absorption of these chemicals when you, for example, take it orally. When you take it orally, to get into the stomach, from the stomach to then to the small intestine, where most of the absorption of these substances takes place, especially the biomolecules when they are broken down into, for example, monosaccharides, amino acids in the case of proteins, and fatty acids in the case of lipids. So we have made mention already that you can have a category of these molecules. They can be classified on the basis of their polarity. You have highly polar and much less polar. So the much less polar are the lipophilic. And we know that when you have a compound that is highly lipophilic, can actually dissolve from the membrane and then transverse the membrane and find itself on the other side of the membrane, from the outer to the inner, inside the intestinal cells, the enterocytes. So basically, when a compound, which is a drug, when a drug compound is more or less lipophilic in nature, it can easily find itself inside. It can be transported inside. So that can increase the chance of bioavailability. We talked about the bioavailability concept. So, and that is that. But when you have a compound that very small molecules, very small. So all these very small molecules, whether they are lipophilic or hydrophilic, in most cases, we know of a simple diffusion process. So they can easily pass through membrane through simple diffusion. They don't have much challenges passing through the membrane. We also have another category that can only pass through maybe through a certain transporter molecules. These ones are usually the ones that pass through these transporter molecules by what we call a facilitated diffusion. And we know what facilitated diffusion is, where you have another thing coming out and then the energy use maybe lead to release of energy. So that energy maybe is going against concentration gradient, and that help facilitate the movement of some of those substances inside. You also have another mechanism that involve active transportation, where some specific transporter molecules, in most cases, we call them efflux transporter. We'll talk about pestering metabolism of xenobiotics, where in pestering we'll be discussing basically on the excretion. So we talked about the influx and the efflux transporters. In this case, we have the influx transporter, basically like ABC, the ATP binding acid. We call them ABC transporters. So mostly these transporters were expressed around the intestine, around the gut generally, but more expressed around the beginning of the intestine, the intestinal mucosa. So they are expressed there, and most of their functions is to help in transporting these substances inside the internal cells. So they require energy. So some compound can only go through into the intestinal cells through these transporter molecules. That is a classical active transportation. But you can also have some set of compound that can go through with the process we call endocytosis. We all know what endocytosis is, and I am very much sure we also are very familiar with some drugs that are encapsulated, and mostly what they used to encapsulate was especially at a very small molecules. You can see they used a highly lipophilic substance shell that can facilitate the movement of these substances. It gives kind of a cover so that those substances can easily be transported inside without having any challenges. So the same thing apply when it comes to dermal route. For example, when you have a substance applied on the skin, or you have a pollutant dropping on your skin, this something happen. This substance can actually diffuse into the cells, the dermal cells, into the dermal cells. Maybe number one, it could be they can go, they can diffuse through the skin cells. Through the skin cells, they pass into the skin cells, they go down, like enter into the vessels before they finally find themselves into the blood vessels. Or you can have this diffusion occurring in between the cells. So in between the cells, more or less the extracellular matrix, and then finally find themselves inside the blood circulation. Or you can have some of them passing through the hair follicles, all the force on the skin down to the blood circulation. Or in the case of inhalation, you have these drugs after having inhaled, they will then find themselves on the alveolar cells of the lungs. And they can undergo the process we made mention depending on the polarity of the substance. Either they can dissolve on the membrane, or they can go through simple diffusion, they can go through precipitated diffusion, they can go through active transportation, the endocytosis, etc. All this can happen. And there are a lot of criteria to determine whether a substance, most especially in the case of drugs, whether a druggable compound or a compound or a substance can likely be considered a drug, most especially when you look at some parameters that has to do with the polarity, the parameters that has to do with how big is a molecule. You know, when the molecule is too big, there is a high chance that it will have hindrances getting absorbed. So polarity also, and what are the contributing parameters, physical parameters of compounds that contribute to this polarity issue. So there are a number of criteria. So when you read, you can come across some of these criteria. Classical of them is Lipinski's rule of 5. For example, for a compound to be considered a druggable compound or to have the potential to be considered a drug, it says that the compound must not have a hydrogen bond donors more than 5, or the molecular hydrogen bond acceptors, it should not be more than 10. Or the molecular weight should not be more than 50. Or the log P should not be also more than 50. The log P is more or less the partition coefficients, which has to do with the ability of this to dissolve in non-polar solvent relative to polar solvent, which is relative to water in this case. There is also a polar surface area in a molecule, which part of the molecule or the surface of the molecule can be regarded as polar. Sometimes called topological surface area. There is a rotatable bond, the solubility, parameters, etc. So there are a number of criteria apart from the limitation of the pipe. There are quite a number of criteria that determine this whether a compound can be considered or not. So, I supposed to talk about distribution first before this first metabolism. So pardon, please let me go down to. So in the case of distribution, you have cases where some of these substances, like we just talked about absorption, and after absorption, you can have this compound getting themselves, if it is through oral, they are inside enterocytes, which are the intestinal cells. So you can have a significant amount of them that were unable to screen out, okay, maybe basically based on some parameters, which majorly has to do with this polarity. So more lipophilic, most lipophilic find themselves inside. So classically, you will see that most of these substances that find themselves inside the cells, so they were finally absorbed. You see that the majority, high amounts of them were lipid, the more lipophilic than the polar ones. So you put that at the back of your mind. So you will mention one or two things that relate to that, and you will see much sense. So once these cells get themselves inside the enterocytes, they need to be taken to the blood. But on their route to the blood, they will have to pass through. The hepatic vein, portal vein, from the hepatic portal vein to the liver, from the liver, and then into the blood. So into the blood, they pass through the extracellular fluid, the blood and the interstitial fluid between the cells, and then finally them into the cells before they exhibit their own function. So in distribution, that's to do with the movement of these substances. And remember, once they get in their way, so many things can happen, and that can reduce the bioavailability of the drugs. From for example, the movement, distribution, and inside the blood. Sometimes some drugs bind to plasma protein, and some does not bind to plasma protein. They move in form of pre-fraction. So pre-fraction, you find out that the drugs that usually bind to plasma protein. So sometimes even when they get to the point of where they need to go into the cells, they may not separate, so they tend to be less bioavailable than the ones that are transported really. Because those that are transported really, they have a higher chance of getting absorbed to various salts as they are passing through the blood cells. So this is basically distribution, that when these substances get into the cells, they need to be distributed. And this is the route of the distribution we mentioned, that from the from the enterocytes, they get into the portal vein, from the portal vein into the liver. And there's quite a number of things happen right from the from their entry into the enterocyte, which we mentioned, because as they are going, their concentration somehow, maybe due to these physical interactions with the tubes, some of them are becoming unavailable. So the bioavailability is also reducing. And at the same time, also, we'll talk about some certain metabolism, which they affect the activity. Okay, so I will go back to first pass metabolism. In first pass metabolism, basically what happened is that at the concentration are actually decreasing somehow, but these drugs are also being metabolized because we're talking about biotransformation, but one thing you need to know that in biotransformation, these reactions takes place in majorly in the liver. We say the reaction majorly takes place in the liver, but that doesn't mean only liver. Some of these enzymes for the some of these enzymes are expressed even in the enterocytes, the intestinal cells. They are expressed there. So all the phase 1, phase 2, and phase 3 receptors or molecules, because most of the phase 3 are rather transporters, but phase 1 and phase 2 enzymes are all being expressed in the intestine. They are also expressed in the kidney. They are also expressed in the liver. They are also expressed in some other organs, and basically many organs, but specifically these 3 organs you mentioned, the expression of these substances is high there. So once the substances get absorbed, it can, the metabolism starts actually. And of course, as the metabolism occurring, the concentration of these things reduce gradually before it reaches the final target. So inside the enterocyte, there will be some little metabolism, and when they get to the liver, also there is a little metabolism. And in some cases, even when they are being transported inside the blood, some of them will get into the red blood cells, and they will definitely be metabolized. So as they are metabolized, the concentration decrease. So the systemic bioavailability now will depend on the fractions of drug of the substance that were absorbed, because not all of them will be absorbed. So sometimes you can even have a very some drugs you can have less than 10% absorption. Some of them less than 20%. Some of them you can even have some that are less than 5% absorption. So you can see that what is absorbed is significant. Is non-significant. So of course, out of it also before reaching the target, some of them will be metabolized and they will not be available. Even though metabolism, what does the metabolism mean? Because the metabolism could mean activation or deactivation. It could mean a lot of things we will discuss. So you have the systemic bioavailability comprising of the fraction absorbed, the fraction that escape metabolism in the enterocytes, the fraction plus the fraction that escape metabolism in the liver, and the fraction that also escape metabolism while transporting to the target tissues. So this is basically what happened in this passive metabolism. If you have any question, please do not hesitate to ask whenever we come for the class. So then we will talk about the biotransformation. Remember, I said we will see through the absorption, distribution, and metabolism. So in biotransformation, basically the biotransformation, the aim of the biotransformation is one, which is like basically the main thing about metabolism of foreign substances. Because once the foreign substances enters the body, unlike the biomolecules that are known by the body, which will definitely go through different pathways, beneficial pathways. But these ones are substances that are not needed by the body. The primary aim here, because the body doesn't differentiate between a pollutant, a food additive, or a drug. You take a drug, with an intention of a good intention that this thing will go and do something I need. But the body look at it as a foreign substance. And what does it do? Okay, you foreign body, or whatever, you are a foreign. Therefore, I need to take you out. I just need to take you out. You are not belong here. We don't recognize you. We don't need you. Therefore, you need to go out. So whatever reaction it can or it will undergo, the reaction is meant to take you out. Remember, we made mention of when these substances are coming in, to some extent, they are being screened to a certain degree, in such a way that you will have majority of them are being lipophilic in nature. So, and for you to excrete these things, there needs to be a level of polarity. They need to be soluble. They need to be polar. And then, so the essence of this biotransformation is meant actually to make these substances applicable for EV excretion. So this is the main aim. So, but in the process, you can have activation or deactivation. It depends. When I say biological activation, let me just say bioactivation in most cases, when we are dealing with a drug, for example, so it's activated to become it. So the biotransformation can lead to activation of the substance that comes in. So it will exhibit its own biological activity. Or the biotransformation can be deactivation as well also. So when it comes in, it's already active. It started doing biological activity. After metabolism, then the metabolism converted to inactive. So it deactivates. Or it can be toxic substance. Most cases of polyton, even though some drugs can also be toxic to nature, but even though preliminary screening, before taking, must be sure that it is not toxic. So, most especially considering the concentration. So, but you can have a polytons that even at a smaller concentration can be toxic. So the process can also mean make it detoxifying, or at the same time could also be activation. Because it can come in non-toxic, but the metabolism can make it toxic. So anyone can take place. But the aim thing is that the aim of these reactions is actually to make it more soluble, therefore it can easily be excreted. Otherwise, without making it soluble, it may not be excreted. So there are pathways. So this biotransformation. You can have some of these compounds that does not even need to pass through any of the reactions. Because remember, we mentioned that, of course, you can have, most especially when you have encapsulation, and this encapsulation facilitates the absorption of quite very polar substances, and the polar substances get them substance. And you can have some of them also getting into simple diffusion, as we mentioned. So, and also facilitated diffusion, as well as the use of ABC transporters. So you can have some of them that are a bit reasonably polar and can easily be excreted. So you can have this set of compounds that does not even need to pass through the phase 1 and phase 2 reaction. So they can directly be excreted easily. Or you can have some that does not need phase 1 reaction, they go to phase 2 reaction. So we talked about the difference between the phase 1 and phase 2. What is the essence? But the aim of all the two is to make it more soluble. How do you make it more soluble? So you have to follow this stepwise before you make it soluble. So let's discuss phase 1 reaction, which will be basically what we'll be discussing in this class. So in phase 1 reaction, like I will say, the aim is actually to make it polar. But how do you make it polar? You make it polar, in most cases, you react it with a substance, or you attach substance that will make it polar. Or you attach a very, sorry, let me put it this way, you attach a very polar substance. So the percentage overall of the molecule will be polar. That is a classical conjugation reaction, which is the base 2 reaction. But for that reaction to take place, first of all, you need to, a kind of, this one reaction is more or less making the molecule easily react with a more polar substance. So it's a kind of activating the compound or making the compound reactive. Or yes, making the compound a bit reactive so that it can undergo the conjugation reaction easily. But you can have some cases where just the P1 reaction is making it polar enough so that it can easily be excreted. So the major reactions in P1 include the hydroxylation, oxidation, reduction, hydrolysis. But there are quite a number like epoxidation, peroxidation, desulfuration, deamination, etc. So you can have this and they don't need to pass through. And in most cases, you will see that the reaction ended up adding either OH group, superhydroxy group, amino group, carboxy group, or even removal of these ones, depending on what will make the compound more soluble. So we've already mentioned that the reaction takes place in plasmic reticulum. In these organs we mentioned, the cells of these organs we mentioned, and we said that the most common reaction is hydroxylation reaction. The hydroxylation reaction catalyzed by cytochrome P450 monooxygenases. But of course, there are other enzymes that are involved, like aldehyde dehydrogenase, alcohol dehydrogenase, and so many of them. And the classical reaction is shown in this reaction equation. You can see we have this RH, which is the Xenobiotic, and you have oxygen, you have a reducing equivalent. What does the enzymes do is it attach, we said it's basically hydroxylation. This enzyme basically catalyze hydroxylation, and it is involved in metabolism of more than 50% of the drugs. More than 50% of this. You can have some other enzymes that are involved in metabolism, but this cytochrome P450 is involved in more than. And out of there are many. Also, we talked about cytochrome P450s now, and let's see how diverse this set of enzymes are, and the potentials to be used in so many other areas. So this is classical. We talked about the classical reaction with the mechanism of cytochrome P450 oxygenase, and I think you must have known this enzyme, mixed function oxidase in fatty acids metabolism. I think fatty acid desaturase. You know fatty acid desaturase, and you know the mechanism. I think in fatty acid metabolism, we talked about desaturation reaction, and you must have done that in 200 level. You know how the desaturation reaction takes place, and that's the mechanism. Sorry. So the mechanism is exactly as it is in this one. So we'll briefly also see the mechanism. So the mechanism, the compound, the enzyme, like we said, is a hemoprotein, and at the same time a flavoprotein. It contains a plavin nucleotide in form of plavin adenine nucleotide, and it also contains a heme group ion. So what happened? So this heme and that is from the prosthetic group, and it uses NADPH as an electron source, and the oxygen as the primary electron. Like we said, is found mostly expressed in the liver, small intestine, and then in some other cases you have them expressed in kidney, lungs, and placenta, mostly placenta. We talked about cases of induction of these enzymes and how this is being expressed in the placenta, most especially in pregnant women, how that can taking some things that induce some of these drugs can cause a problem with pregnant women, specifically when I talked about that. So what happened in this reaction is that the NADPH plus H sub at the electron source, and they got oxidized, and the plugin nucleotide will accept these electrons, and then finally the electrons that are used to reduce iron from iron 3 to iron 2. And this process leads to the transfer of the proton to one molecule of splitting of oxygen molecule, of which the two proteins will attached to the one molecule of oxygen from water, while the other atom of oxygen, as a molecule instead of atom. So splitting of the one molecule of oxygen to form one atom will react with the proton to form a water, while the other atom will be attached to the Xenobiotics. So initially it's ROH, now it became ROH. So the ROH now can easily undergo reaction for conjugation. Or if the OH is enough to make the molecule polar, then it can easily be excreted. This is basically the mechanism of cytochrome P450 monooxygenase. So before we see some characteristic of this cytochrome P450 monooxygenase. Number one, it is a widely distributed across different species. So this enzyme can be found in many species, microorganisms, bacteria, or I say microorganisms. So generally, they can be found in microorganisms, fungi, algae, etc. Number two, they exist in a large number of isoforms. You have a lot of isoforms of this chemical. That is the reason why the classical way of naming them was developed, in which you have the cytochrome, which the word you can see example here. You have Cyp1E2, Cyp2C9, Cyp. This is all to differentiate between a different the isoform of this proteins. So, sorry, they are they are classified on the, or they are differentiated on the basis of amino acid sequence similarity. So first of all of them were the cytochrome P450 monooxygenases. But how do you separate this to say, I belong to this family, this I belong to this family? This was done on the basis of amino acid sequence similarities. So anyone, for example, there are many, up to about 150 or more. So you can have some group that have about 40% amino acid similarity sequence. So this group were classified in one family, for example, and a number is given to the family, say an Arabic number 1. So this one belongs to family 1. So you can also have another one that have this 40 up to 40% similarity sequence in the amino acid sequence. They are also given, let's say, another family 2. Like this, you have a lot of family. They were divided into family. So in each family also now, you say, okay, all of you have at least similarity of about 40%. You can have some among them that their similarity exceed that 40%. So if the similarity within the group exists maybe above 55%, so they are also further subdivided into subfamily. So, for example, you have 10 that share a particular sequence similarity in the amino acid sequence. You classified them into family 1. So this family 1, within them, there are some, because we didn't say only 40%, 40 and above. So only within them, you can have some that their similarity is even more than 40. It's up to 50, some up to 55, some above like this. So anyone that the similarity sequence is above 55, they are classified as subfamily. And they were given the alphabet. So you can have, you are still in family 1, you can be E, some can be B, some can be C like this. You can have 1E, 1B, or 1C. And further also, the Arabic alphabet is given to each of these enzymes. So you are given a family 1, subfamily E. In subfamily E, they are now arbitrarily given a number. So in subfamily E, you are number 1, you are number 2, you are number 3, like this, like this, like this. So they identify as number 1. You have the Cytochrome P450, which also you are belong to the Cytochrome P450 oxygenase as CYP. And the 1A2, for example, as indicated here, indicate that this belongs to the family 1, subfamily A, and is number 2 in subfamily E. Likewise for all of them. I hope that explanation is enough for you to understand what. So they are, this is how they are represented with all this code. So, and you can have this code, if written in italics, it indicates the gene coding for that particular enzyme, but not in italic indicate the enzyme itself. The next thing is that this set of enzymes have an overlapping substrate specificity. This in particular explains why this compound can actually metabolize thousands of compounds. You don't know where the compound will come from. Some of them are from food industries, some of them pharmaceutical industries, some of them chemical industries. Just come in contact with them and they become their substrate. Now, remember we talked about specificity in enzyme chemistry, where we know there is a enzyme being very specific. So they only catalyze their particular substrate. But now you have some enzymes that they have ability to catalyze, to metabolize a structurally different compounds. So this particular property is even used most especially like in environmental biochemistry, bioremediation specifically. You can isolate an enzyme from a microorganism that can be able to metabolize a lot of pollutant in the environment. So you can take a waste for example, this the hydrocarbon from oil producing is a contaminated land with all these petroleum products that are from diverse compounds. So this can convert them to harmless substances. And basically, so many other applications can make use of this peculiarity of these enzymes. Now, the next thing is that these enzymes are inducible, meaning that some substance can increase their activity or can decrease their activity. Inducing this can mean, or increasing their activity could mean increasing the synthesis of the messenger RNA. That is the transcription. So it induced transcription, so more messenger RNA are produced. And you know, when more messenger RNA are produced, what does that mean? It means that there will be more translation because the messenger RNA will be transported to the cytoplasm that to the ribosomes and translated to produce the proteins. And once the proteins are produced, then they will perform their function. So you can have some substance that can trigger this messenger RNA synthesis. Or it could also be stabilization of the messenger RNA, because some messenger RNA are very level. So once they produce a lot of small thing can lead to degradation of them. Or stabilization of the enzyme itself after being produced. Or some of them could even be could lead to inhibition of this enzyme. So they can be induced and they can also be inhibited. We'll see and that explains the concept of drug interaction. You can take a drug and it works for you perfectly. But you are taking the drug and something came up and you also need to take another drug separately, different from this one. Not knowing that maybe there is an interaction between this new drug you started taking with the drug that is metabolizing the first drug you're taking. So if that happens, the interaction different. If the interaction means is induction, meaning it will induce the expression of these enzymes that is involved in the metabolism of the second drug. For example, the first one induce the expression of the enzyme that involved in the metabolism of the second one. So you are now taking the first one and it is working for you. Not knowing that at the same time also it induced the expression of another enzyme. I'm talking about the Cytochrome P450. So it is working, and at the same time increasing the expression. It doesn't matter, you don't have any problem if you don't need to take the second drug. But by the time you now have a reason to take eventually there is a reason for you to take the second drug. Now you are taking the second drug. You're given a prescription of a particular concentration. There is possibility that when you take that drug, you may not function well, or you may not see the effect. Previously, you've been taking the same maybe dose, it is working for you, but because now you are taking another drug, that drug is not working for you, simply because it's induced the production of the enzyme that's involved in the metabolism of that drug. And what does that mean? It simply means that as that drug comes in, there are a lot of enzymes that can metabolize it with a short period of time. So the duration it would take in your system, of which the time it would take before it got completely metabolized and get out of the body, is needed and targeted based on the concentration given you. So that time, that duration of action, it will reach that time in your blood for it to be able to perform its own function. But because the expression of these enzymes that are involved in the metabolism of it is high, so once it comes, it quickly gets metabolized and easily excreted within a short period of time. So it doesn't take much time in your system. Therefore, it does not have enough time for it to carry out the function as it's supposed to do. Therefore, it easily gets out of the body. Or you can have a case where you have an inhibition. So it leads to inhibition of the enzyme. If the first drug leads to inhibition of the second, the enzymes involved in the second drug. So when you take the second drug, for example, you take, for example, 20 milligram, and the 20 milligram is doing for you. You've been using it and it's okay for you. But now that you are taking it alongside another drug, or you started another drug before, now another reason for you to take that drug comes, and you are taking the drug now at the same 20 milligram concentration, and you discover that it's giving you some side effect, it's giving you some difficulties like this. Yeah, simply because the concentration, that 20 milligram, is staying, for example, when it's supposed to take 8 hours to easily get out of the body. Now it's taking 24 hours in your system. So you have all the 24 hours the drug is working. then you can have some side effects associated to being the drug being delayed in your system for long. So this is the classical drug interaction. That is why it is important to know which drug is given, which drug you need to be given, so that you will not have a potentiation or you will not have an inhibition of activity. Well, classically, see some of these examples. And the last one, we say that Cytochrome P450 exists in polymorphic form. You can have with the polymorphic form a particular enzyme. Some individual express a polymorphic form of a particular enzyme in such a way that you can have a polymorphic form being high in activity, or you can have a polymorphic form exhibiting a low activity. So you have an individual that, if I with respect to a particular drug, when I take paracetamol, I usually have a really bad headache. But another person, when taking paracetamol, will tell you that I've been taking paracetamol, but I don't feel anything. Paracetamol has never worked on me. Or a particular drug worked for me, but this is my friend, it doesn't work for him. But probably it could be your enzyme is a polymorphic form. Maybe the polymorphic form of the enzyme that is involved in the metabolism of that drug is the catalytic activity, for example, is very low. So when you take the drug, it may not work well for you. For example, let me give this example of individuals taking this Indian herbs. You have an individual taking Indian herbs, and this Indian herb is being catalyzed by cytochrome P450 in your system. If you have a friend who is taking these things, I know you must have one or two discussion, and even I'm not saying you, but you are taking, but you can have friends that are involved in all these things. And if you have friends, you will know the kind of discussion they do, most especially sometimes trying to mock themselves or trying to mock a friend who is unable to take enough of these things. Or you will take small, somebody will go high for a long period of time, or you will go high for a short period of time. So it's, to them, it's something they used to even be proud of. Like, I can take more of this, of which if you do, you spend days under the influence of the drug. So they feel like it's a kind of is a bravery to take as much as 80 milligrams. And within a short period of time, you are out of it. You are no longer under the influence of it, for example. And you have now another person that can take 20 milligrams and will be under the influence of this thing for the next 24 hours. Not knowing that it is actually genetic and it could be related to polymorphism. So some individuals, they will be under the influence of that drug because simply the enzymes have a low activity. Therefore, that thing you take would stay longer period of time in your system, being metabolized gradually, before it finally metabolize completely and get out of your body. While others will have a polymorphic form, a genetic isoform of the drug, of the enzyme, that can quickly metabolize it and get it out of your body. So the influence, you will see this one, the duration of action will be high, the longer will be elongated, while the other one, the duration of action will be shortened, and the drug can easily get out of the body without taking much time. So this is basically, so I have given an example of some reactions in phase 1. I've mentioned hydrolysis. We can see aspirin, acetyl salicylic acid can be hydrolyzed to form salicylic acid and acetic acid. And we can see atropine can be hydrolyzed to tropic acid or tropine. Induction, we can see butyric acid to glutamic acid, nitrobenzene to aminobenzene, and we also see oxidation like benzene to phenol, gets called ethanol to acetic acid, etc., etc. So this is just an explanation I give with respect to induction and inhibition of Cytochrome P450 which I mentioned that some xenobiotics have induction, synthesis, while some inhibitors. This is an example of drugs. For example, we say you can have an individual who is taking cigarette. Who is taking a cigarette. And that person eventually is now to take a drug, theophylline, which will be metabolized by cytochrome 1 into, maybe due to a lung problem, I mentioned that has to do with the quality reason or something like that. And you are taking 20 milligram. And when you take that 20 milligram, it will work for you. You will have relief. Your chest, your breathing, the difficulty in breathing will drop. Now suddenly you started taking a cigarette. Because the cigarette smoke, some chemicals inside the cigarette smoke are inducers of cytochrome 180. Therefore, the expiration of cytochrome 180 will go high. Remember, I mentioned the model of the induction, which I said it could be the increase in the expressions of the messenger RNA, or it could be stabilization of the messenger RNA or stabilization of the protein. So it can induce this cytochrome 180. And when it increases, it simply says that at that time range when the level of cytochrome 180 is high and the need for you to take theophylline comes, you have a difficulty in breathing. When you go and take theophylline, that 20 milligram you used to take, it may not work for you. Why? Because the expiration level of cytochrome 180 is high. So once theophylline comes, it can easily be excreted within a short period and it will go out of the body. Meanwhile, maybe the 20 milligram will stay for 24 hours in your system or 28 hours in your system. And because of the level of cytochrome 180 is high, now it's taking just 8 hours or 6 hours and get out of the body, and you will not feel the action of it. Or you can be taken at the same time. For example, they say, okay, you're not taking cigarette, or rather, you take another antibiotic, erythromycin. You are taking theophylline, or you are taking erythromycin. You are under erythromycin, and then now after a week of taking erythromycin, now you have to take theophylline because of difficulty of breathing. So the erythromycin is an inhibitor of cytochrome. So it inhibits cytochrome 180, which drastically reduces the activity of the enzyme. So even when you take theophylline, 20 milligram which initially work for you perfectly, but now it tend to be like you are overdosing. Can give you some adverse effects of overdose. Too much of it or something like that. And this has to do with inhibition by the activity reduced so that it cannot be cleared within the 48 hours. Rather, it will remain in your system for 3 days or 4 days before it is cleared. That is given this is. So this is basically the induction and inhibition of cytochrome P450 monooxygenases, and something applies to all of them. They are inducible enzymes, and they also have inhibitors. So I think, yes, this is the end of this class. So please, just like I said, I was in the class and you guys are not. There are some where you didn't even listen. I decided to record the class and share with it. Like, unfortunately, I wanted to do this the day I entered the class again because of too much things I was unable to until today. So please take your time before the next class, which will come. I don't know if I may need to present myself on the Tuesday so that you will also decide and be sure of when to have the class on Thursday. And if it is possible, and if it is not possible, then we'll have to find a way also. See, maybe Wednesday or somewhere, I will fix the class. So please take your time, go through this and all the explanation. If you have any question, keep your question, put it somewhere written when we come for the class next week or the next class, which will start phase 2. Reactions and the examples, please you come up with all your questions regarding this, and I will give a summary also of this so that we proceed to the next. Thank you very much.